everyone and welcome to this new and exciting lecture on M13 Phage vectors that I'm going to deal today. For those of you who understand Hindi better, I've already made this video in Hindi, so you can check out that video. For those of you who understand English better, so this is the video that you can watch. Apart from that, for our new viewers, I would like to tell you that I have already made, or I am in the process of making a series on the different types of vectors, and I have covered multiple vectors in that series. So this would be the new lecture in that series. So stay tuned, and what all we would be discussing in this particular lecture, let me just briefly tell you. So we would be discussing about what is M13 phage, what is the composition of M13 phage, different diagrams that are related to it, applications of M13 phage, and how do you identify that a particular M13 phage is having your gene of interest or not. That means basically genetic engineering applications or implications of M13 phage vector. That is also what we are going to discuss in today's lecture. So without much ado, let us start. So phage M13 vectors, now first point, used for obtaining single strand copies of cloned DNA, especially suited for DNA sequencing. Now, a very important point that I would want to mention over here for this particular point is that M13 phage, this is actually a virus. So you would be knowing that phages, when I say phage, so the first word that you are seeing over here, it is phage. What does phage means? It is referring to bacteriophage. Bacteriophage. Bacteriophages are viruses that are known for attacking bacteria and killing them. So this is also a type of bacteriophage, and we are talking about this phage because this phage works as a vector. That means it can be used for transferring our gene of interest inside the bacteria. So this is a type of a virus that would be helpful for transferring our gene of interest inside the bacteria. So now, coming to the genomic composition of this virus, so M13 is basically a single stranded virus. That means, that is why I have written over here that it is used for, this M13 phage is used for obtaining single stranded copies of cloned DNA. Now when I mention cloned DNA, what do you understand by cloned DNA? Cloned DNA simply means that when you are inserting your gene of interest inside a particular vector, then that is called as clone. For example, let me just draw it over here for you. So suppose this was the, let us say the vector DNA, and now if I am adding my gene of interest, for instance, I am showing my gene of interest blue in color, and what is going to happen? This gene of interest, or this my gene of interest, would be integrating between the vector DNA, something like this, So you can see that this is from the vector, this is from the vector, and this is my gene of interest. So it will become cloned DNA. So when we are interested in obtaining single stranded copies of the cloned DNA, then we make use of M13 phage, primarily because M13 phage vector is also a single stranded virus, or its genomic composition is single stranded DNA. Now coming to the next point, and apart from that, one more important thing is, because it is used for obtaining single stranded copies of the DNA, it is very useful for DNA sequencing. Now you would be, if you have a little bit of idea about DNA sequencing, you would be understanding that in case of DNA sequencing, we require single stranded DNAs. Even if the DNA is double stranded, what happens is the DNA gets denatured, or it divides, or it breaks down into two separate strands, and then sequencing starts. Therefore, since over here we are obtaining single stranded DNA copies, then therefore the DNA sequencing becomes far more easier. Now, derived from 6.4 kb genome of E. coli filamentous bacteriophage M13, or F1. In the first point only, I told you that this particular vector is called as M13. An important addition to the information is that it also can be named as F1. So either you can call it as M13 vector, or you can call it as F1 vector. Both are the same things. Now again, M13 or F1, both of these bacteriophages are present or can attack E. coli. So that is why I have written that they are bacteriophages of E. coli. And the genome size, the rough genome size of M13 or F1 bacteriophage is 6.4 kb genome. Now, whenever you are studying vectors, the most important question that is being asked in recent trends is about the genome size of the vector. Apart from that, there are questions that are related to what would be the genome insert or insert size in case of the vectors. So this is one of the information that you should remember. Next is phage has single stranded linear DNA genome. This is again what I have already told you. So when I said that the phage M13 phage is single stranded in nature, so this is what I meant. It is again written the same thing has been mentioned over here also. Now, an important point that I want to cover over here is that although M13 phage is single stranded, but when it enters inside E. coli, obviously I said that it is a vector or it is a phage of E. coli, so it would be attacking E. coli cells. When it attacks E. coli cells and enters inside the E. coli, it starts replicating and then it changes its single-stranded form into double-stranded circular form. Now, this is very important and very crucial and asked in many life science examinations. So, if the phage is outside a particular cell, then you can say that it would be having single-stranded form, but when it is inside a cell or it has attacked an E. coli, then definitely it would be present inside double-stranded circular form inside the E. coli. So this is the second thing that I would want to tell you. Now you can take a quick photograph of this and then we can move forward. So coming to the next slide, this is just the diagrammatic representation of the M13 genome. This basically is the genome of M13 phage vector. There are multiple things that I would want to highlight over here. So first thing first, let me just choose a color. Now you can see numbering over here. So these are the different genes that are present on the M13 genome. Now these are the genes numbered. You can see that some of the numbering has been, or some of the annotations have been done over here. For example, the second gene over here is for DNA endonucleases. It is used for double-stranded replicative form. I have already told you that when the M13 phage enters inside the E. coli, the single-stranded DNA form converts into the double-stranded replicative form. So this is the gene that would be responsible for the same. The next one is the 10th number, and it is a DNA replicative single-stranded circle. So it is used for, this particular gene is used for the DNA replication. Gene number 5, single-stranded DNA binding protein. This is a gene that would be producing SSB, single-stranded binding proteins. Number 3, basically up till here, that means 6, 3, 8 and 7. These are capsid proteins. Capsid, what does it mean? Capsid is the capsids are basically a proteinaceous layer that is surrounding a virus. So these genes would be responsible for synthesizing the capsid. Capsid, one more important thing, that it is made up of capsomeres. So this is also an important information. Apart from that, this is the origin of replication, ORI. I would be talking more about ORI as we progress in our lecture. Now this is also one of the annotations that is given over here. There are different DNA, the different genes and what are the functions that has been mentioned over here. You can check it out over here, but it's not that important. So those that were important I've already mentioned above over here. A little more information has been mentioned over here. Now let us move on to the next slide. Quickly you can take a photograph of this so that we can move ahead. Okay. Moving on to the next diagrammatic representation, you can see that this is the electron micrograph of filamentous M13 phage. This means that this is how M13 phage virus would be looking inside the electron microscope. But this is a normal diagrammatic representation. In this, some of the annotation has also been done. You can see that multiple proteins, the name of multiple proteins has been mentioned over here. For example, G3P, G8P, G7P and G9P. Now actually this end over here, this end is the fixed end. That means it is basically attached, and this end is the free end. So G7P, G9P proteins are present at the free end, whereas the G3P proteins are present on the attached end, and importantly G8 protein are present throughout the body of the M13 phage. This is also an important information that you should know. So you can take a photograph of this and then we can move further. Okay, moving on to the next slide, let us move on. Only infects F plus and F prime cells. This is again an important information. In my previous videos, I have already told you what is a F plus cell and what is an F prime cell. But for your understanding, let me just quickly revisit the concept. So, if the F plasmid, let us say that this is a bacterial cell, this is a bacterial cell, and inside a bacterial cell, if F plasmid is present, so this is the F plasmid. So if F plasmid is present, then the cell would be called as F plus. If the F plasmid is absent, then it would be called as F minus. Okay. Now coming to the concept of F prime cells. What do you mean by F prime? Understand this. Suppose, let us say that this was the plasmid inside a bacteria. I am drawing everything inside a bacteria over here. Let me just draw the bacteria also. Okay, so this is the bacteria and this is the F plasmid. Okay, now definitely you would be knowing that the inside a bacteria a chromosome is necessary. That means the genome of the bacteria is definitely necessary, and plasmid can be present and also cannot be present. That means the plasmid is not necessarily present always inside the bacteria. So what I am drawing is that this is the genome and this is the F plasmid inside a bacteria. What happens is at times this F plasmid would be integrating inside this genome. So F plasmid would be integrating inside the genome. Let me just draw it quickly over here. So suppose this was the genome and F plasmid quickly integrates inside it, something like this. Like this, you can see that this was the genome, the blue one, and the F plasmid has integrated over here. Now, if this is the condition, then this condition is called as HFR condition, high frequency condition. But at times, what happens is after a while, this F plasmid decides that it would come out of the chromosome. Now, when it decides to come out, at times it does not come out very clean. So when it does not come out clean, it often brings the chromosomal genome or chromosome genes along with it. For example, like this. So if I write A gene over here, B gene over here, so you can see that when the F plasmid is coming out of the genome, of the chromosome of the bacteria, it does not come out clean, and along with it, it brings out some of the genes present on the chromosome. So this condition is called as F prime condition. Okay, so considering this, we can say that the M13 phage vector would only be infecting the F prime cells and F prime cells and F plus cells. Now, F pili is used to inject the 6407 nucleotide genome. Now, as I have already said, that M13 phage vectors, they are responsible for infecting the bacteria. Now, in order to infect the bacteria, they have to transfer their own genome inside the bacteria. Now, what is the method through which they transfer the genome inside the bacteria? They utilize F pili. This is the structure through which they transfer their genome, and the size of the genome is 6407 nucleotide, and this genome is then transferred inside the bacteria, and then this genome would be taking over the entire machinery of the bacteria, and so on and so forth. Now, double-stranded replication form of M13 would be forming. As I've already said that initially it was in single-stranded form, but when it enters inside the bacteria, it forms the replicative structure and double-stranded circular form is produced. Again, coming to the concept, how does replication takes place inside a bacteria? So there are two methods through which replication takes place inside bacteria. One is the theta method, and one is the rolling circle method, that is also called as sigma method of replication. Over here we are talking about the sigma method or rolling circle mechanism. So when the M13 phage vector affects the E. coli, then rolling circle mechanism takes place, and because of this the duplication of the genetic material of the M13 phage occurs, right? Now, each infected E. coli has 100 copies of the M13 genome. What does this mean? So, in general, we can say that if a bacteria is present, then approximately 100 M13 phage vectors can reside inside a bacteria. This is the capacity of the E. coli. But when the replication starts and the phage starts multiplying inside the bacteria, it is seen that 1000 new phage particles produced per generation. That means after each generation, thousands of phage particles starts coming out of the bacteria. So this is again a testimony to the fast speed of the replication of virus inside that bacteria. So you can take a quick photograph of this and then we can move forward. Okay. Now phage M13 does not lyse the infected host cell. This is the another point that I would want to mention. M13 phage does not believe in doing the lysing cycle. You would be knowing that there are two types of cycles present, or there are two types of cycles generally implemented by the phage. One is lytic cycle and one is the lysogenic cycle. So that is what is being mentioned, that lysis is not relative to M13 phage. So M13 phage does not cause lysis of the bacteria but forms turbid plaques due to growth retardation of these cells. Now what happens is, the bacterial cells inside which the M13 phage has caused infection, now these bacterial cells will encounter a decrease in the growth. So the growth would be so much decreased that you would see an abnormal structural difference with the other normal copies over here. So the copies, the structure of the copies, the structure of the colonies of those bacteria would be different as compared to the other normal bacteria. Why? Because the growth would be so much retarded. Why the growth would be retarded? Because there is so much of load of M13 phage inside those bacteria, which is not allowing the normal growth to progress. So that is why we are calling it as turbid plaques would occur. This is the word that is being used. Next is double-stranded form of M13 genome for producing recombinant molecules. What does this mean? Now, double-stranded form of M13 genome for producing recombinant molecules. Now, what happens is, because when the vector is going inside the bacteria, it is having your gene of interest also. So it has become recombinant. Why? Because there was a part of the DNA of the vector, and your gene of interest was also present over there. So that is why we are calling it as double-stranded form of M13 genome is having recombinant molecule. That it is a recombinant molecule because your gene of interest is also present over there. Now, single-stranded form of M13 genome is used to obtain single-stranded copies of DNA insert vectors inside the media. Now, let us understand this. There is an important thing. Now, when you are doing the experiment and when you are allowing the M13 phage vectors to enter or to attack the bacteria, so that your gene of interest is transferred inside the bacteria, understand this, all the M13 phage would not be able to attack E. coli. There would be certain M13 phages that would go inside the E. coli, and there would be certain M13 phages that would not be able to go inside the E. coli, and they would be present inside the media only. Now those M13 phages that are present inside the media, they would be having the single-stranded DNA. Whereas those that have gone inside the bacteria, they would be producing replicative forms, double-stranded circular colony. That means if you want to isolate, or if you want to find out or isolate the single-stranded DNA, you can check out the media from where you can take out the M13 phages and take out the single-stranded copy. Then if you are interested in the double-stranded one, you can find out the cells, the bacterial cells, and from there you can take out the M13 phages. They would be having the circular DNA. This is what is being said over here. Now, you can take a photograph of this and then we can move forward. Okay, so this is a diagrammatic representation that is basically showing the preparation of single-stranded M13 DNA from an infected culture of bacteria. Understand this, that let us say that this is a culture of infected cells. That means there are certain bacteria that are present over here, and there are certain M13 phages that are present over here that have not caused the infection. Together they are present inside a tube. Okay. Now when you do the centrifugation, what will happen? The bacterial cells will centrifuge down. These are the pelleted cells, and these would be the M13 phage suspensions, right? This would be the M13 phages. These would be the bacterial cells that would have settled down. Now what you do is add polyethylene glycol to the phage suspension centrifuge. Now this would be M13 phage suspension, because M13 vectors are present in this suspension. It would be taken out over here, and then polyethylene glycol would be added to this. When you are adding polyethylene glycol, what will happen? Then you would be getting the M13 phages and resuspend the phage in the buffer. Now you would use a buffer in order to resuspend the phages. Now you take out the DNA by using the phenol chloroform method, right? So when you are adding phenol chloroform, what happens is the M13 DNA would come out in the chloroform form, or phenol form is present in downwards, and protein is present just intermediate between the two layers. So this is the protein form, this is the M13 DNA, this is the phenol. Now we take out the M13 DNA, remove aqueous layer, that means aqueous layer is basically of chloroform, and ethanol is added. Now when you add ethanol, you get the M13 DNA, and then you can resuspend M13 DNA in a small amount of buffer. In this way, you can get the M13 DNA. But since this DNA would be in the single stranded form, these were the M13 phages that have not entered inside the bacteria. So therefore, you would be getting the single stranded form of M13 DNA from the infected culture of bacteria. So this is the entire method that I had talked about previously also, and this is a diagrammatic representation of this. So this diagram is present in T.A. Brown, Wiley and Blackwell publications. Now, you can take a photograph of this and then we can move forward. Okay. Now, selection of recombinant DNA. This is very easy, and you might have read about this. We are going to talk about the blue-white screening colony method over here. So DNA inserts plus 507 base pair of non-coding region. Now understand this, that there are the DNA insert means the gene of interest that you want to transfer inside the bacteria. This is the DNA insert plus 507 base pair of non-coding region. Now in the M13 phage, there is a region that is called as non-coding region, but it has ORI region also. This non-coding region is having the ORI region, and the size of this is 507 base pair. Now polylinker, this is the name of a structure. This is a structure basically. Polylinker has an important function. I would be talking about that in just a bit. Polylinker is used, and the size of the polylinker is 42 base pair. It has several unique restriction sites. Now polylinker, the function that polylinker plays is that it is a sequence, not that big a sequence, just 42 base pair sequence, but it has multiple restriction sites. What do you mean by multiple restriction sites? That means restriction enzymes. Multiple different types of restriction enzymes can come over here and cleave the polylinker, right? So this polylinker is used, and several unique restriction sites is placed within the LAC-Z alpha gene. Now remember this, that this polylinker is added in between the LAC-Z alpha gene. For example, let us say that this was the LAC-Z alpha gene. This is the entire LAC-Z alpha gene, and in between the LAC-Z alpha gene, what we do is we place our polylinker. Now this is our polylinker, right? Now this LAC-Z alpha gene, what is the function of LAC-Z alpha gene? You might have heard about the LAC-Z gene. LAC-Z gene is basically utilized for coding the beta galactosidase enzyme. Beta galactosidase enzyme. So LAC-Z gene is used for coding beta galactosidase enzyme, and LAC-Z alpha, specifically alpha, is used for encoding the amino terminal. Now you would be knowing that all the proteins have an amino terminal and a carboxy terminal. So the amino terminal of beta galactosidase is basically coded by this LAC-Z alpha gene. Okay, hopefully you would have understood this. Let us move forward. You can take a photograph of this. Okay, now understand this, that this polylinker basically was used because it has multiple restriction sites, and our gene of interest would be now present or coded, or it would be placed inside the polylinker. What do I mean? Let me just draw over here. So I said that this is a polylinker, or let me say that this is the LAC-Z alpha gene, LAC-Z alpha gene, LAC-Z alpha gene, and this is a polylinker. This is a polylinker, and utilizing this polylinker, because I've said that this polylinker has multiple restriction sites, utilizing the multiple restriction sites inside the polylinker, we can put our gene of interest somewhere over here. So this is my gene of interest. Okay, so this is your polylinker and this is your gene of interest. So this is what we are talking about. Okay, now understand this vector plus IPTG plus XGAL. I've already said that, just a moment. Yeah, so I've already said that this particular vector, we can check out whether our gene of interest is present inside the vector or not through the blue-white screening process. Now this is the vector, we are using IPTG. IPTG, what is it? It is the inducer. What will it induce? It will induce the production of beta galactosidase gene. So if IPTG is present, more amount of beta galactosidase gene will be produced. Okay. Now what is XGAL? XGAL is a compound that when interacts with beta galactosidase enzyme, it produces blue colored colonies. So over here we have put XGAL also. Now in case when there is a vector, you are adding IPTG, you are adding XGAL, and suddenly you find blue colored colonies. What does this mean? Remember, if you find blue colored colonies, then you can conclude that your gene of interest is not present over there. That means your gene of interest could not be attached at the site of the polylinker. Why? Because in this case only the LAC-Z alpha gene would be intact. When the LAC-Z alpha gene would be intact, the alpha site or alpha region of the beta galactosidase would be formed. When beta galactosidase is formed, as I said, when beta galactosidase interact with the XGAL, then only the blue colored colonies are produced. So if blue colored colonies are produced, then you can say that our gene of interest was not present inside the vector, right? That means the vector was non-recombinant. Our gene of interest was not present over there. But when you are using vector, DNA vector, and DNA insert is present, IPTG plus XGAL, the colonies would be colored. The colonies would be colorless. Why? Because this DNA insert would be present inside the polylinker, and because of the polylinker, because of the the presence of the gene of interest, the XGAL would be disrupted. The XGAL would be disrupted. So, the LAC-Z would be disrupted. So this LAC-Z would be disrupted because of the presence of the gene of interest. And when that happens, what will happen? The beta galactosidase would not be produced. So beta galactosidase would not be produced. And when beta galactosidase is not produced, it cannot interact with XGAL. If it cannot interact with XGAL, automatically the colonies would be colorless. So over here blue colored colony, over here colorless colony. This is called as blue-white screening. Now same strategy is applied for puck plasmid also. We would talk about puck plasmid. We have already talked about puck plasmid previously. Now examples, what are the different examples of M13 phages? M13MP8, this is one of the example, M13MP9. And what is the difference between these M13 phages is that there is a slight differences in the LAC-Z alpha gene. This is the basic difference. Next, restriction sites were created in them using the synthetic polylinker sequence. Now gradually as research progress, what is being done is restriction sites are being created in these using synthetic polylinkers. Already previously made polylinkers are being added to these M13 phages, and now what we are getting is M13MP2, Previously, the M13 MP2, that was one of the first M13 phage vectors, is now being converted into M13 MP7. And this M13 MP7 has pre-positioned Eco R1 site, BAM H1 site, SAL1 site, and PST1 site. So this was one of the problems previously. We had to add polylinkers before, but now as the research is progressing, the previous M13 vectors are now converted into the modern ones, which already have these restriction sites in them. So this is again a great step in case of vector biology. So now what are the features? Talking about the features. Sorry, I did not give you time to take the photograph. You can take a photograph. Okay. Now let us move on to the features. Very large inserts can be cloned, of course. The sizes can be very large, can be cloned, since packaging does not depend upon the genome size as in case of lambda vectors. Now in case of lambda vectors, basically there is a constraint of packaging, but over here there is no constraint, so therefore large size or large insert size can be cloned. Pure single-stranded copies of double-stranded DNA inserts are obtained in abundance. Of course, double-stranded as well as single-stranded copies can be obtained. I've already told you this. Now, DNA inserts are accepted in either of the orientations. Some recombinant clones will produce single-stranded copies of the complementary strand of DNA insert. Now understand this, that you would be knowing that, just a moment, some recombinant clones would produce single-stranded copies of the complementary strand of DNA. Now, let us say that M13, we started by saying that M13 phage is a single-stranded phage, or is a single-stranded vector. But when it enters inside E. coli, it duplicates and then forms double-stranded form. So this is a double-stranded form of, let us say, M13 phage. Although it is a circular form, but for your convenience or your understanding, I am drawing it in linear form. Now, when the phage would be coming out, that means when suppose this is a double-stranded phage that is present inside the bacteria. Now the phages would start multiplying and start coming out of the bacteria, right? When they come out, some of the recombinant phages might have this strand and some of them might have this strand. This is what is being said. So some recombinant clones will produce single strand copies of the complementary strand of DNA insert. That means if this was our DNA insert over here, so complementary to this DNA insert, let me just draw it with a different color, then you might be able to understand it. Now, suppose suppose this is the M13 phage DNA that was present inside the vector initially. When the vector was not going, or when the vector was not present inside the bacteria, it was present in this form, and this is the gene of interest. But when the replication would be taking place after this M13 phage enters inside the bacteria, the complementary form of this gene of interest would also be produced. So this is on the single strand, but when it goes inside the bacteria, it would be producing a complementary strand. So that is what is being said, that when the phages would come out of E. coli, some of the phages might have the complementary strand, some of the phages might have the original. This is what is being said. Phage particles in a single plaque would have same single strand. If you are talking about a single plaque, a single region, right, where one group of bacteria would be present, all of these would be producing the phage particles with a single kind of strand. So if in this, let us talk about, if this is a plaque and inside this plaque there are multiple, let us say, bacteria, and they are producing M13 phages. So all the M13 phages would be having would possess the same single strand. They would not possess the complementary one, they would be having the same one. But if you are talking about another plaque, they might, or this might have the complementary strand instead. Right? This is what is being said. Now, helps in precise DNA sequencing. I have already told you how it helps in DNA sequencing, so you would have got a clear idea. Now, bacterial cells, oh sorry, you can take a photograph of this. Let us move on to the next one. Bacterial cells remain viable. Now I have already said that it does not form the light, it does not progress through the lytic cycle, therefore the bacterial cells remain viable, they do not die, although the growth is limited. Next is recombinant DNA is obtained within stable bacteriophage particle. So I've already told you recombinant DNA is obtained. Why am I calling it recombinant? Because it is having the vector as well as the gene of interest. So since they are from different sources, that is why it is called as recombinant DNA. Is obtained within stable bacteriophage particle. When the it is bacteriophage, inside the bacteriophage it would be present, the recombinant DNA is present inside a stable bacteriophage. This is what is being said. So this is all. I believe you would have got a clear understanding of what I taught in this particular lecture. Apart from that, a very important information is that our GATE classes, just a moment. A very important information that I would want to share with you is that our GATE classes have started running from the 19th of June. That is a little while, some time back. If you are interested in joining these classes, then you can contact us 8299134528. You can get the detailed video about this batch on the top, and you can check out that video, or you can contact us through this number. We can help you out to enroll. So hopefully you would have liked the lecture. If you liked the lecture, just hit the like button, subscribe to our channel and share it with other friends as well. Thank you. Have a good day.